So this live session for the course Functional Genomics. I'm sure all of you are going through the videos and these lectures and the links that we provide. And I'm hopeful that you find this course useful, easier to follow, and then you are able to appreciate the new, concept called functional genomics. We try to make it as easy and as interesting as possible by giving the most updated content directly taken from many of the research articles. So what we will do now, we'll take up some of those questions that you may find it difficult to understand or would like to know about it. So my suggestion would be, let us not take anything too technical a question, because the more detailed technique related questions if you send us, we will be able to direct you to appropriate reading materials, because that would be time consuming to explain in the live session. But we will take up questions that are of more generic, and many would be benefited by the interaction session. So you can write to us in the chat box your questions, and we'll be able to address them one after the other. At least you can introduce about you. That way we would know how many of you are there and what is your background, and it gives you a kind of understanding for us to answer your questions.
I think one should start writing their query in the chat box, so that we know that what are the questions you have, and then we can answer it. And okay, welcome Gitanjali, and
nice to see your note, Gitanjali, and it's great. I mean, to see that professionals who are already teachers in various colleges and other places are attending this course, and we find it very, very gratifying. And of course, you can go about asking your query. Yeah.
Okay, so her query is not, my query is not course related, as in theory part of it. Probably she is writing that what she wants to.
Sure, please go ahead. You can write. Yeah.
Okay, so Gitanjali is asking, I'm trying to register for exam, but for many days the site is under maintenance. Okay, so yes, Gitanjali, there was problem with the server, and soon it will be resolved and you will be notified, and if it will take time, then definitely the registration time will be extended for the exam enrollment. And for that, you do not worry, that will be taken care. That is NPTEL team is working on it. So another Lata Kumar Swami. So again for sir, that
yes. Anything that related to the course and assignments and extension and so on, I would recommend you please write in the forum, and our office, like Rashmi and others, as well as from the NPTEL office, will take care of your queries and they'll address to you. So that should not be any issue at all. So you please, we will, because these are specific queries, and we will address them. Certainly we will address them.
Okay, so Lata Kumar Swami. So this short course was very informative. I'm curious to know more about DNA sequencing biosensor.
I certainly want to elaborate what you mean by DNA sequencing biosensor. So you may want to write a little more detail on that. Exactly what is that you are referring to as DNA sequencing biosensor? Okay.
And then Rajnikant, is this course applicable for Career Advancement Scheme UGC?
Yes, that is, it is part of it. It is one of the course recognized by the UGC file.
Yeah.
Right, yeah. Certainly, I would encourage you to take that. Exams, soon it will open. That is already been answered. You will get all those, isn't it? So there will be...
Yeah, the NPTEL team is working on it,
So,
and we have communicated your queries already, your questions to them, and they said that soon it will be resolved, and if necessary, then they'll take care for all this. So you do not worry. Then Rashmi, again, that the course is very helpful for.
Yeah, thank you. Thanks for enrolling in this course. I'm sure this course is of help to you as well, as a teacher. That would be great. Yeah, if you can give your feedback on what more topics can be covered as a teacher. Then we can think of in the next version there could be some revisions added to this course. And there is Periyasamy, and he's also a professor in Vellore Medical College. What way this course is useful to medicine field? Absolutely. I mean, any advancement in genomics has great relevance to medicine, because the future medicine is going to be what is called as what people call is personalized medicine, meaning the medicine designed, tailored to individual genomic makeup. In that way, the genomic tools that come up, either when you talk about genome sequencing or expression profile or variations, these are areas in which now there are various attempts to link that to the way you react to certain infection, or the way you are susceptible to certain diseases or resistant to certain diseases. So that sort of gives you an idea as to, even before you are exposed to certain pathogen, what we hope that you are sort of, understand based on your genomic sequence, what are your, kind of susceptibility. So therefore one can have a preventive measures. That's what's called as future personalized medicine. So that's something that's going to come up. So in that way, the genomic tools are very, very important in terms of how the disease can be managed, prevented, or diagnosed, or treated. So it's a future. So certainly it's an important area where it should be integrated with the medicine as well. So there is another question by Govind Srinivasan, a student from Trivandrum, is talking about the genome editing technology. How is the double nicking technique different from HDR? He is asking very, very specific questions. What we will do is that we will connect you with the relevant material, because it requires some schematics and other understanding. So if I explain now, so that would, can you reply to him in this text itself, sir?
Hi Govind. We'll, for this we have already discussed in the slide that how this NHEJ and HDR, by creating double, NHEJ is a double nick technique one, and how HDR is different from that. And we'll give you more details on that, because here we do not have a schematic from where we can explain you. So we'll be providing you the links for that.
Right. So what I suggest, Govind, is that if you could send the same query in our forum, so we'll connect you with the relevant material, more detailed material, so you can go through them and understand. Okay. So
Okay.
then there is Latha Kumaraswamy. There is one more note.
Rashmi.
Rashmi, sir, can you please elaborate more on the reverse genetics more appropriate than forward genetics in modeling human disease? Okay. I wouldn't say this is modeling human genome. I would say it is modeling human disorders. That's more specific way, because in the sense that what we do in the reverse genetics is that you create similar kind of mutation in a genome of a model organism, the mutation that you find in the human, and then see now that what happens to that particular animal, and then you sort of recreate the disease to the extent that is possible by generating very similar kind of mutations. That's why reverse genetics is far more useful in terms of human disorders. But forward genetics is something to understand, when you don't know, it's the natural mutation equivalent process. You create mutations randomly and look for certain phenotypes and try to map where such genes that contribute to a particular function is localized, what kind of protein they code, and so on. So that's where the forward genetics is. So especially when you know the mutation in human genetic disorders, you go back and then recreate that mutation in the animal. So that's where the reverse genetics is far more useful. Okay.
Yeah, Lata, I'm working as assistant prof. The course was very informative to update myself. So.
Very nice. Again, I would request you share with us your experience. Even if you find that there are some topics that you would like to be covered, you please share with us. We'll try to collate them and then come up with a better course, maybe 1 year down the line.
Okay, Govind, thanks for your understanding, and definitely we'll be providing you the reading material for that again.
Right.
And then Geetanjali sir, can you tell us recent example where CRISPR-Cas9 technique is used?
Well, there are large number of papers. In fact, every day papers are being published. There is a site called PubMed. That is I don't know how many of you are aware. There is a site called NCBI, meaning National Center for Biotechnology Information. This is maintained by the National Institute of Health, USA, and in that there is a link called PubMed. So it lists all the papers that are published in the area of biomedical sciences. So if you go to PubMed and there is a query box, and if you give this particular whichever query that you would like to sort of search, and if you say CRISPR-Cas and then you give, it will list in an order that is the most recent paper would come first, followed by even the very first paper that reported this. So all these papers will be there, and you can see that every other day then you will have one or other publications coming up with this kind of informative articles. So certainly you can go through.
And I think in week 3 tutorials also we have covered how to search that. How to use PubMed over NCBI.
Absolutely, that is also there. Yeah. So what other? Without schema, it would be not provide much understanding to you. Okay, that is your writing. Sir, can you tell us any recent example? That's what we have explained to you. Right. Maybe for this one of the articles also can be linked. You can give the link there itself, maybe if you want. Yeah. So what we'll do is, for the earlier question, there was a question about
how this double nicking is different.
Yeah. So since there are two questions, one on a specific process by which the genome editing takes place, and then there's another query on what are the new papers that site reports, and we can also search here.
Yes.
So what we are doing is we are giving you links right away, so you can see that that link already we shared with you that talks about the papers that give you more detailed understanding on that. So you can have a look at it. Um, just want to know more about
A type of ethical aspect that
right.
under CRISPR editing technique.
Right, right. So the ethical aspect, there are two different aspects. One is that when you use this approach for the model systems like the mouse, rat, and you know chimpanzee or whatever models that people normally study, um, that's a different kind of ethical approach. There, it is allowed to when you want to create certain mutations that are and known to change the gene function, you certainly can create them, but there are committees that has the guidelines for each country, and then the committee goes through them and then approve these such kind of a research protocol. That is when it comes to the human. You can also give the PubMed link. But when it comes to the human, then it is it's a different ball game. So it is not that for exploratory purpose, the committee would never allow any such um, uh, research to be conducted on the human. So it is only when you know that this approach perhaps can save the life of an individual who is suffering from certain disorder, then possibly a committee may consider this approach to be applicable, right? So that's what we call as clinical trials. So that's again, it's a very elaborate process, and that is only when you know that there are no other ways by which you can save a patient, but this approach is very promising. Then you can possibly it can be attempted, then it can happen. But again, you cannot use this approach to correct everything in every single cell of your body. So it's a then there are issues. So certainly only in certain conditions such approach can be used, and this is being considered. There are a lot of newspaper articles that are there, so you can see that. I mean, it depends on what conditions that you are looking at. Yeah.
Okay, Vedyasamy, is it possible to use functional genomics in human diabetes patients by means of any of the specific method of functional genomics?
Um, yeah, certainly. For example, when you talk about metagenomics, people talk about um, biomarkers. Even when you look into your gut microbiome, it sort of tells you. Now there are many papers which sort of looks at your microbiome in your gut as to an indicator of your metabolic status, and then when that's destabilized, and then may have a certain effect. So now there are large number of papers which sort of talks about your skin microbiome with your health, your gut microbiome with your health, and so on. So these are again functional genomic approaches, because there are no other way by which you can type the microbe that are present in your body. So you basically sequence the genome and try to using the next generation sequencing, and then classify the microbes and then try to find an association. So it's got a very good diagnostic value coming, but how that would influence the physiology is something still people are studying. Whether it is a cause or a consequence, there are debate, and either way there are supports. These are next stages, but it looks like there are clear correlation with regard to the kind of microbiome that is there in your body with your physiological status. So even when you talk about diabetes, certainly this is one method of functional genomics, which maybe eventually it will become a very good tool to identify your metabolic status without really monitoring for a long time.
Yes, and we have shared the link here as well for CRISPR-Cas9 and HDR versus the double nicking, and also for the PubMed search.
Yes, the PubMed search, again the link, you can click that, and then whatever query that you wish to add, you can add and search, and it will tell you what papers. Yeah.
Yes, I think if you have more queries, you can write it there, because Mhm. CRISPR-based genome-edited plant animals are permitted in farming because genetically modified, specifically transgenic plants are facing lot of issue in India.
Yes, there are two different things. One is that whether this kind of approach is permitted in plants. For large scale, there are large number of groups that do this kind of experiments. So for you to understand. But when it comes to, you talk about genetically modified plant, this is for the commercial scale. For example, you have developed certain things in the lab scale and you find that is very beneficial, either drought resistant, pest resistant, or it gives products that have longer shelf life or the yield increases, then you would like to take it to a commercial field. That has a large number of issues. There are regulatory bodies that would approve. Once it is approved, then it also depends on various other factors at times, because anything that is commercial, there are companies and there are people who oppose commercialization because there are also angles like, for example, the seed is something that only the company can give. That is one common sort of issues raised against the genetically modified um, plants, because normally the traditional farmers, they seed and they grow and then keep part of the yield as a seed for the next crop. And whether such kind of thing would happen, because the company may say that no, it will not work and things like that, because they also have their own commercial interest. So these are all beyond the science. So there are many other factors that influence, but certainly it is not something that it is not allowed. So it is not. It is certainly it is allowed. There are many instances where it's being tested and people are using it, trying to get a better yield variety and so on. Yeah, certainly it is possible and being done. Yeah.
Seems everyone has they got their answers for their queries.
Any other uh,
Any other query?
query? Well, thanks, Pariswamy. This is very good news to hear that you plan to take up research-based genomic studies, and it's wonderful. Um, and that's the way to go forward. So, um, so if that course really helped in understanding many like you, then that's the ultimate um, aim of this course. So I'm very, very happy and hope that you would go through many more such courses in the future as well. Yeah. Well, there is one query that there could be a WhatsApp group for the post-supportive study material. Well, I mean, I don't know. If you know all other students who are participants of this course, if you'd like to do it at your own end, possibly yes. But given that courses could have hundreds of students, I don't know whether such kind of things are possible. We will not be able to officially do that, because what we do is on the forum that we pretty much do the same thing. But if you find some study links, study information that is present elsewhere, we would like to share with others, I mean, feel free to post it on that, on the
Google Forum.
yeah, Google Forum, so that will be helpful to others as well.
Another limitation with WhatsApp is if multiple messages are coming, then one, I mean, probably will miss the queries from somebody. So in Google Forum, it's easy to track it. So therefore, we prefer to have a Google Forum.
That's right, yeah. So the other question is, can we have some useful links on these topics or some other paper? So what we try to do is, we try to give the required, information, right, for the course. If you give too many links, I mean, of course, for any topic you have more advanced papers, we can always share with you. But if you give that to everybody, some may find it very, very hard. So what we give as part of our release of, the video lectures every week, we give the essential information, the links that have the essential information that are supportive to these lectures. But if you are interested in having an understanding of a given topic in more depth, you write to us, we'll be able to share with you. So that cannot be sort of, told to everybody, because then it becomes difficult, because we are going to have students, participants who are just doing masters, or somebody who is already teaching, who are professionals or medical professionals. So we have heterogeneous audience here. Therefore, giving more advanced links would be a burden for others. So, but however, if somebody is interested in knowing more, you are most welcome to write to us in the forum. Then we will share with you the links for a given topic. So that would be more appropriate. Yeah.
As sir said, and already if somebody asks that they want a bit more information on that particular given topic, we regularly share the recent articles.
Right, absolutely. The another query is,
Okay.
you have group for
In continuation of that WhatsApp.
yes, I mean, this is something that you can start one. I do not see why it cannot be done, but certainly amongst the participant, if you would like to open a WhatsApp group, and so we have nothing against it. I mean, you please go ahead and make it. But the only thing is that it would be difficult for us to be part of it and attend to it. So that's all I are saying, right? Yeah. Is the exam in November going to be MCQ type? Yes, whenever the exam, that would be announced. But yes, these are multiple choice question type, all are objective type questions. Yeah. The question is, the non-nucleated cells like RBC or any nuclear metal like RNA, DNA can be used for genomic studies. Well, it depends on what you are trying to look at.
Normally when we take blood sample particularly, so then we take WBCs, because we can RBCs we can remove from hypotonic buffer or something, and then you can take WBCs, and then from there you can isolate DNA or RNA, because whenever you will be collecting blood, it will be whole components will be there, and then that can be further used for genomics.
Right, your question is on the RBCs, whether they would have any nuclear. Obviously it doesn't have much of the DNA, but whether they have the RNA, well, it could be, we don't know. As you know now, there are a large number of studies which suggest that even in your circulatory system and in many other places, you have these vesicles that carry RNA, right? That like regulatory RNA, which one or the other species, and of course there are studies which is suggesting that there are trace amount of RNA that are, people call it residual RNA and so on, that is there. So it is there. You would see, for example, in later studies where we talk about non-invasive procedures for detecting certain prenatal conditions of a baby that are still within the mother, people use the serum and the blood and so on. Likewise, there are many vesicles that could go and talk with RBC, which may have, give a little bit of RNA there. So obviously, if at all there is something which will be very, very low, that's what I would comment here. So whether it is something that is planned that way, or accidental, these are all, further studies may confirm. Right now, there are reports which suggest that the RBC has some small amount of RNA. Yeah.
Gitanjali, sir, can we also include a small session on the connection of genomics to various other omics?
Sure, that we'll consider when we revise this course next time. So we would certainly consider. It's a good suggestion. So we'll consider that here.
And then Govind, in future, can CRISPR ever be used as a regular medical treatment for patients having genetic diseases, or is it still going to remain as a last resort treatment, provided the clinical trials are safe?
Yes. So it's possible. Let's look at this way scenario. Like in the beginning, when the egg cell and sperm were isolated, when people were trying to fertilize the egg in a test tube, it was more of an lab-based method to understand how the development takes place. That's how it was developed. And then soon they started using this for individuals or people who have had difficulty in having child. So now we have what is called as test tube baby, which is nothing but in vitro fertilization. Say every other town and cities have these clinics now, wherein they sort of mix the sperm and egg to make this what is called the in vitro fertilization. So to begin with, this was a lab-based approach. Similarly, when we know that these approaches will not have any harmful effect elsewhere, that sort of understood and certified, maybe this also would eventually become like the test tube or IVF clinics, for example. So think of anybody who is having beta thalassemia or any other inherited disorders, then they may have this the sperm egg collector, and then they may fix this error and then allow that to fertilize, and you could have babies in which the genetic alterations have been converted into the wild type, therefore they become normal, otherwise they would have had the disease. So it is a possibility. Most of these approaches to begin with have come from lab studies, and now they are into clinics, so one may anticipate such things happens in the future here.
Okay, so next is from Janvi. Can you talk about junk DNA, which is not really a junk?
Well, it is like this. When we did not understand something, that time it was called a junk. Okay, so we need to understand in evolution, nothing that doesn't have any function would stay true. So we all have seen, for example, if you subscribe to this concept that all the humans have evolved from monkeys or the primates, then we lost the fur coating like we had in our body, and we also lost many of the uniqueness of the monkeys that they had, including tail and so on. And possibly these are lost because for certain conditions, when things change, there are certain conditions that is no longer essential. So those that are not essential would be lost in time when evolution happens. Therefore, that applies even to your genome, right? You can see in our own anatomy, there is a small portion of your intestine called the appendix. Now, people develop certain problems, then surgically it is removed, and then you become all right. So what you call as appendix is something that we call them as vestigial organs that no longer functional, but they were functional, for example, in many of the other mammalian species. So therefore you lose it because they are no longer functional. So same applies to the genome as well. If the junk DNA, so-called junk DNA, is irrelevant for the genome to function, we would have lost it. We did not lose it, and still we have them, is itself a proof that they are not just junk. They have certain function that we did not understand. That's what is changing now. Now we know that the non-coding region of your genome is involved in various functions. The function is need not be exactly like the coding sequence. You have certain nucleotide sequence. It can be any sequence. There could be variation within individuals, variation between individuals, variation between different races, populations, but still they are doing their function. We know now that the way the chromatin remodel, the way the chromatin, like we know that in every cell of the 23 or 46 chromosomes, they have certain regions where they are localized. So all these possibly are regulated by the so-called junk DNA. That is one. And second, they are involved in, because they make large number of these so-called non-coding RNAs, which have great regulatory roles. So we are trying to understand how they contribute to the way the genome functions. So maybe 20 years, 30 years from now, you're going to have a new course on junk DNAs, possibly because you'll have that much of information from this particular section of the genome. So it comes about that, yeah. So I think we are beginning to understand now. That's what it is. That's why I say it is not junk, because if it is junk, then it would have been deleted a long time back. Just like you have in your laptop or desktop, there is a trash can. So as you add more files, one day you go and clear everything. So that would have happened if it really is junk. That's not the case.
I think in continuation only, can you highlight on long non-coding RNA for disease prediction?
Well, again, there are, so you have to understand that, the one thing that you need to understand is that any DNA sequence that are repetitive in nature becomes very, very difficult to analyze. Most of these sequencing data that we get, these are all curated data, in the sense that you isolate RNA, you sort of sequence the RNA using next gen sequencing, and then you sort of curate. So when you do a curation, then you basically, most of the other repeat containing sequences and other things are removed, and then processed RNA is what sequence what people consider. If there are such long non-coding RNAs that have repeats in containing RNAs, when you don't know the exact function of it, more often these are sort of discarded because you really don't know what they are. So, but even then, when people look into two different disease states, like one disease condition, other one is a normal condition, and if you have means to have the tissue and RNA, one can do this kind of sequencing and try to find out the difference. But what you need to understand here is that even if you take two individuals, same tissue, both are normal, then you would find differences in the DNA, the RNA sequence, what kind of species that are there. These are individual variations. So unless you have really a good number of samples and you have looked at all of them and sort of you have applied some statistic tool to see the differences that are significant or not, one cannot really correlate such kind of expression. So these are the challenges, because when you look at humans, and this being applied in many conditions now, people are trying to see what are the differences in the transcriptome when I compare it to the normal and the affected. So that's anyway are there, and there are many papers. So if you send a query on the discussion forum, then we'll be able to share with you.
So another one, Govind Srinivas. Is there any drastic impact of CRISPR to the gene pool of a species, assuming the modification done is just for treat a disease?
Well, theoretically speaking, it shouldn't, right? You are just correcting one error which happened because of one reason or the other, and you are correcting that sequence to a sequence that is present in the majority of the population. So it should not really make a huge difference to the pool of the species. That is assuming whatever you have done is restricted to only that particular site, which we believe the CRISPR does, right? So if that is the case, yes, there should not be much of an impact. But if there are some changes, modifications, it could be epigenetic, others which still we don't know, a long-term effect of such kind of alterations, whether it affects the way the genome functions. So that would, time would tell. But as of now, theoretically speaking, assuming that this change happens only in the given site, then it should not have major impact on the species.
Okay, so Rashmi, sir, what about bar bodies? These also have any such hidden or silenced gene information. How do they get inherited?
Yeah, bar bodies are nothing but the very compact X chromosome in females, because there are two X chromosomes in females, all the mammalian species, of which one of the X gets inactivated. So we believe that is just to compensate the difference in the gene dosage. The male has one X chromosome, female has two. So if you have both X chromosomes are active, then for any gene that is present in the X chromosome, there will be twice the amount of product as compared to the male. That's where we believe that evolution went with this mechanism of randomly inactivating one of the X chromosomes in the human female, and that is what was identified as to begin with, was identified as bar bodies. Possibly the cats, that's where the first were identified in the female, and that is named after that scientist, if I recall correctly. So that's what it is. So it is basically, it is a what is called as heterochromatinized X chromosome, one of the two X chromosomes. Yeah. And then certainly it is inherited, because the X chromosomes, when you have meiosis, these two X chromosomes are sorted to two different X cells, and obviously both become active and goes on.
Okay. Janvi, why animal tissue culture is not used as primitive stage then directly using model organism?
Tissue culture is not used as primitives. Yes, they are being used. It depends on what you would like to look at. Say, for example, if I am interested in understanding how a protein is being made from a gene and how the protein is modified, translated, modified, localized within a cell, then I use the tissue culture to understand all this phenomenon, because this cannot be done using models. So it depends on what questions that you're asking. And if you are trying to understand a function of a protein in terms of the whole organism, then it has to be at the model organism. So the tissue will not, for example, I'm talking about whether removing a gene or modifying a gene would make that individual to be more susceptible for a disease, let's say. This can't be modeled in cell lines, because disease susceptibility cannot be studied in cell culture. It has to be, you have to have an individual, an organism. So therefore it has to be looked at organism. So it all depends on what is the question that you are asking, and that's where it is. Yeah. So there's nothing called primitive or advanced. It depends on the question, right? What the research questions, right?
And then Pidasami, that blood group antigen present on RBCs was decided by the genes. How this process takes place in RBC, because RBC is anucleated. But in the primitive earlier stages, the RBCs will be nucleated. As it develops, then only the nucleus will be extinct.
Right, yeah. So it is a process. As the maturation process, these are removed. Yeah.
Most of the queries have come for this CRISPR-Cas9 technique, so it seems that the students
sure, we'll share with you more recent review articles on CRISPR-Cas. I think many of you are interested in that topic. We'll certainly share more information on that. Yeah.
think no more questions are coming. It seems all queries got answered, and if there are more queries, you please write to us. Otherwise, if there are no more queries, then we can end this session.
Well, I mean, it is not that you need to ask this question only using this live session, but you're most welcome to send us in the forum whatever queries that you have. We'll be able to answer all of them to your satisfaction. So that would also help others who haven't joined the live sessions in a way that encourages others to read more on these topics as well. So keep sending your queries on this topic, and we'll be really happy to give more informations, links, reading materials would be of benefit to you. Yeah.
And definitely, whosoever has appeared here, it was nice to see your suggestions and appreciate impact of personalized medicine.
Well, again, this is an emerging concept when you talk about the allopathy, Western medicine. So far, the allopathy or Western medicine looked at the patients as a group, not as an individual. So therefore, now it is being, looked at in a very different way. That's what personalized medicine. So it's based on, for example, your one major factor there is your genome, right? So that's what one major difference between one individual to the other. So if there are some inherent genomic differences, and the differences are known to contribute to certain physiological state, either resistance or susceptibility or higher risk of developing something, then the personalized medicine comes into force even before you develop any ailment, because when you are born, you have your genome is sequenced, and if you have that much of information as to how that can be read and understood as to what are your risk factors, then the personalized medicine comes into force. Then if they say, for example, you have increased chance of developing diabetes, then from early stage onwards, even before you develop diabetes, you can change your lifestyle. Therefore, you can sort of delay or prevent the onset of diabetes. Likewise, if you have a higher chance of developing high blood pressure, then you could, the doctor could advise you your lifestyle change such that you don't get into that, that you take care of food, you take care of your lifestyle. So that really helps you to delay or minimize or prevent the onset of such kind of ailment. Like whether it is allergy, whether it is arthritis, all these can basically, your lifestyle even before the onset of the disease, if you change, it might, extend your normal lifespan in the sense that you are disease free. So that is what is a personalized, the first part, that is preventive. Then you have predictive, that is prediction, and participate in the sense that there's called as 4I or whatever principle. So you basically work with the doctor to prevent or delay onset of this disease. So it is a long-term process. So it starts in what you call as a pre-symptomatic stage. You are at a higher risk of developing disease, yet to develop the disease, but we know that you are at higher risk, therefore you start practicing certain measures that would prevent or delay the onset. So that is what personalized medicine. So one way by which it can be predicted is your genome sequence. The other way could be epigenome that may come, because it is also not only the genomic sequence variation, but the lifestyle of your parents might affect the way you respond to your disease that is coming up right now. So that's another field that is coming up. So we haven't understood exactly how does it happen, but that is considered as epigenetic modification. So that's another field, but that's something. But these are not very new to the traditional Indian medicine. So if you have seen the traditional Indian medicine, that is how it is practiced. But anyway, that is no longer being practiced. But it is coming in the way of Western medicine. Yeah.
And this personalized medicine and more of genetic things, that another course run by that human molecular genetics, that probably it will be run in upcoming NPTEL
Right.
courses.
So the next January there will be, or Feb, there will be there will be courses on, one of the courses is human molecular genetics. So those who are interested may credit that course as well.
And in that, personalized medicine is discussed in detail as well. And another one will be probably scientific communication, so one can join that as well. And then Gaurav, can you please send more information on next gen sequencing and data analysis? Okay, Gaurav, we'll share you the links, because it needs server and then the computational infrastructure. So that's not possible to explain it here. So definitely we'll share you the links from which suggest that how to do the NGS data analysis.
Yeah, we'll share with you the details. Yeah.
And with that, thank you for your explanation. Okay.
So these queries can also be added in the forum. So therefore, whenever we share a response, it helps the others as well. Yeah.
Right.
You're good, and thank all of you. We thank all of you for joining in this live session, and we hope you found this useful. And we plan to have one more session sometime later. We'll share with you the details. Therefore, you can interact once again and understand what you expect and what we can give more. I guess there are no more questions, so I thank all of you once again for joining today, and we will have a great week ahead. Okay, bye bye.